Hello. All right. So this little video is to help you with some of the common terms used in genetics. Hopefully this is mostly a review. So here we go. All right. Gene versus allele. So I like to think of gene as the big category, right? So you could have the gene for hair color. You could have the gene for height. You could have the gene for blood type. And the... Why do I always have to do this? See, even at home I have technical difficulties. Okay, so the allele is a specific version of a gene. So gene is the big category. Like hair color. And you could have the blonde allele, the black allele, the red allele, the brown allele, right? We have lots of variation. And when we teach genetics, sometimes we keep it pretty simple, and even things like hair color are encoded by multiple genes. But for the purpose of defining these genetic terms, okay, allele is the specific version of the gene. Okay? And alleles, the alleles you have are going to determine your phenotype. And what I want you to realize is that you always and only have 2 alleles per gene, right? So you get 1 allele from your mom, 1 allele from your dad. That makes 2 total. So here, shown on these homologous chromosomes, right, this could be mom and dad. This flower has a purple allele and a white allele. Okay. Homologous pairs mean they're coming together and they have the similar genetic information, but they're not identical. Okay. Locus here tells you location. Think of it like an address. And as we get into molecular biology, genes actually have very specific addresses, right? So your neighbors live at one locus, you live at another locus. All together in your neighborhood, you have lots of loci. Loci is the plural. Okay. And that's describing where, on which chromosome, that gene, that allele is located. So down here, I want you to see we are representing genetic information by letters. So letters are our representation. You can use any letter. Whoops. For instance, I like to use big A, little a, big G, little g is good in this, big B, big B. I hate letters that look the same, like C and W and little w, because you got to make sure they look different so you know which alleles you're working with. This figure demonstrates, again, different alleles on different loci, right? So the G gene, the G alleles are located on this chromosome up here, and the W alleles, the W gene, is located on this chromosome down here. And hopefully you can see that these are homologous pairs because they encode the same kind of information. They're not identical. These, let me erase this, are sisters, and the sisters are identical, right? Because sisters have gone through DNA replication, so they're identical. But the ones you get from your mom and the ones you get from your dad are not necessarily identical. So, letters represent the alleles of a specific gene that a specific organism has, and we're only going to be working with diploid, which remember means your chromosomes are in pairs, which means you have 2 alleles of every gene. Only 2. Okay? Now, they might be the same. Homo in science means same, and hetero means different. So an organism that has the same alleles, such as big A, is called homozygous. Tells you they're the same. An organism that has different alleles, say big A, little a, means that they're heterozygous. You can be homozygous dominant, and we'll talk about dominant recessive in just a minute, or an organism could be homozygous recessive. Okay? So, homozygous in a story problem will tell you that for that gene, the alleles are the same. Heterozygous will tell you for that gene, the alleles are different. The story problem may have to tell you that the organism has the homozygous dominant phenotype or the homozygous recessive phenotype. We'll talk about genotype, phenotype, I think, in the next slide. But that's telling you also about the alleles, about the letter representation for those genes. Heterozygous always means that the 2 alleles are different. Now, we're going to start off with regular old dominant recessive, okay? Honestly, a lot of genetic information isn't simply dominant recessive, but we like to start there. And that's where we're going in week 3, so in another week. Dominant is represented by a capital letter, such as big A. Recessive is represented by a small letter, like lowercase a. Okay. Dominant means that one, let's do it this way. As long as there is one dominant, like capital A allele, the dominant phenotype, which we're getting into in a minute, dominant phenotype is seen. Okay, so we'll talk about what phenotype and genotype mean in a minute. Recessive means that the dominant covers it up. So in order to see the recessive phenotype, it has to be homozygous. I don't know why this is going in and out of color. Must be homozygous recessive. So that's illustrated in this picture right here. So in this picture it is telling you, and honestly this is not the way eye color is inherited, but we're going to pretend that brown eyes are dominant to blue eyes. Okay? So if you have someone who's homozygous dominant, and they mate with someone who is homozygous, homozygous recessive, okay, all their offspring, that looks funny, these are the gametes. We'll get to this in class on Tuesday. All their offspring are going to look dominant. Okay, because oh, sorry, I guess I should have been here. This homozygous dominant parent and this homozygous recessive parent are all going to have heterozygous offspring, and with just one capital B, you get the dominant phenotype. So you can't see blue eyes unless someone is homozygous recessive for it. What I want you to see is that you see dominant brown eye color if you're homozygous dominant or heterozygous. Okay. Note, with heterozygous, we don't add dominant recessive because heterozygous implies one of each. Okay. So take a look at that. Make sure you really understand what this dominant recessive means, and that you understand the difference between genotype and phenotype. So, genotype is your genetic information. And if you look at somebody, you really can't tell what their genotype is. Okay? If things were as simple as dominant recessive, we could look at the class and go, okay, everybody with blue eyes, you're homozygous recessive, but everybody with brown eyes, we don't know. And of course we know there's green eyes and there's different shades of brown and all that good. So genotype to phenotype. Phenotype is your physical characteristics, what you see in a person. Characteristics. So just keep this physical with phenotype. Genotype is your genetics, right? So we represent genotypes with those allele letters, like big A, little a. We represent phenotype with descriptive words, like blue eyes, brown eyes. What I want you to make sure you understand is that you cannot tell genotype necessarily from phenotype. Okay? So if we're talking about regular old dominant recessive, okay, a dominant phenotype can come from multiple genotypes. Okay, so in this case they're telling you that dark is the dominant phenotype, right? And big A or big A, little a, two different genotypes is going to give the same phenotype, right? So if you see a dark butterfly, you cannot tell its genotype. You would have to do some mating to determine that. For recessive genes, the phenotype does tell you the genotype. But what I want you to remember is, again, we will do genetics problems with these simple big A, big B, little b, but that's not really how it happens in humans and all organisms, right? There's lots of genes that affect your phenotype. So, phenotype is the description. Genotype is the genetics represented by letters that represent the alleles of the gene. Alright. Because what we want to get to is we want to look at offspring. So these are two examples of Punnett squares. And Punnett squares allow you to predict offspring given parent genotypes, or sometimes given parent phenotypes, and you have to figure out the genotypes. Okay. So we'll go through Punnett squares in class, but one of the terms I'll use a lot, or actually two of the terms, is monohybrid and dihybrid crosses. So a cross means we take a male and we take a female and we combine their gametes, random fertilization. So Punnett squares are showing you random fertilization, all the possible offsprings that you could produce. One of the things I wanted, a question a student brought up, which is a really good one, is even if you have, like we did, like 3 genes, right? And I told you there's like potentially 22,000 genes that could recombine in different combinations during meiosis. Okay, so this set, we would say 2 by 2 by 2 by 2 equals 8 possible gametes. What I want you to remember is, yes, you still only can produce 4 possible gametes, and in females it's really only 1 gamete. But these are all the possible combinations. Okay. So you can start to think of how this really affects genetic diversity in offspring. Okay, so just because you can make that offspring, or I mean that gamete, sorry, doesn't mean it will happen. What we're trying to do with Punnett squares and crosses is to predict all the possible outcomes. This is important if you're doing breeding. So maybe you're a dog or a horse breeder and you want to not waste your time with animals that aren't going to potentially produce the type of offspring you want. Okay. Right now, currently with humans, we are not allowing them to pick their offspring, though we could talk about this as an ethical issue in genetics. But the power of genetics and knowing both parents' genetic makeup is you can predict the chances of getting certain phenotypes in an offspring. Okay, so let's go back to monohybrid and dihybrid crosses. Monohybrid, mono means 1, di means 2. Okay, I only use these terms when we're crossing heterozygotes. Okay. So if I say it's a dihybrid cross, it means we have 2 genes and both, what do I want to say? And they're genetically heterozygous for both genes. Okay, so in this case, big R, big Y, little I. Monohybrid, 1 gene, heterozygous for both, heterozygous for that gene, for both parents. Sometimes it gets confusing. Sometimes books just say, oh, it's 2 genes. I'm telling you how I will always use it. Dihybrid means 2 genes, and hybrid means different. Okay, so you have to be different for both genes. Monohybrid, 1 gene, and different. Right? And we look at gametes from the parents, and we look at offspring in the Punnett square. And we will come to that in class. Few more definitions. When we talk about crosses, again, mating, sometimes you will see the term reciprocal. Okay, reciprocal means we change phenotypes, or maybe we can say you exchange phenotypes for males and females. This isn't really important for autosomal traits, which is what we're going to talk about first. But when we come to sex-linked traits, it's really important because they're inherited differently. So what I'm trying to show you here is that with these lovely flies, in this case, mom has red eyes and dad has white eyes. So the reciprocal cross would be to change that phenotype, and now mom has white eyes and dad has red eyes. And what I want you to see is that affects the offspring differently. So reciprocal crosses are very important for sex-linked traits. And in our class, we'll primarily focus on X-linked traits. And that's chapter 4. Whoa. All right. Guess PowerPoint thinks I'm done. Okay, another type of cross is called a back cross. And a back cross means you made an offspring with a parent. Okay, so notice we're doing this with wheat. Okay, a lot of these things that we talk about in classical genetics obviously is not ethical to do with humans, right? So we're not going to ask, yeah, let's not even go there. All right, so offspring with a parent. So what this diagram is just trying to show you is, here's the parents, RR, RR, little r, big R, big R. We had this little r offspring. We decided to back cross it to the big R parent. Okay, or here we had a big R, little r offspring. We'll talk about F1 in just a minute. We decided, did I do that? Oh, I forgot that slide. I will put that in with the parent. Okay, so back cross just means you cross it back to a previous generation. And finally, test cross. The reason we use a test cross is to determine the genotype of a dominant phenotype. Right? So remember I said dominant phenotypes, you can't always tell what the genotype is. So if purple is dominant, you don't know if this flower is homozygous dominant or heterozygous because they're going to look the same. So a test cross is always to a homozygous recessive organism. Okay? Always. So for these flowers, little p is the homozygous recessive. Okay. And you do this cross because it will show you in the offspring that if the dominant phenotype was due to a homozygous dominant genotype, you're going to get, in this case, all dominant looking offspring. But if, due to the law of segregation, the dominant phenotype is due to a heterozygous genotype, then you're going to get 50-50 dominant and recessive. Take some time and look at this because you will have problems that will ask you to do a test cross, or that you will have problems that say, how would you determine that, how would you determine the genotype of a dominant phenotype? You always do a test cross. So now, let me see if I can add a slide because I just realized that, so Sorry, you have to see me go through my slow-mo. Okay, wait, I have a mouse. I can do anything with a mouse. I cannot do anything with touchscreens. Alright. I forgot to talk about generations. Because you will see problems that say P, F1, F2. And I'm going to tell you, I'm going to be lazy and write it as F1 and F2. Okay? So P stands for the parental generation. So what we are starting with. Okay? So let's say mom was big A, little a, and dad... Okay, I just said that wrong. Let's say mom was homozygous dominant, big A, and dad was homozygous recessive, little a. And they mated. Okay, and so the alleles that they can produce when we do the law of segregation is we split those alleles. And if we did a Punnett square, really what you come out with is that all the first generation are heterozygous, right? Because dad can only give, or sorry, mom can only give a big A, and dad can only give a little a. Okay, here's the creepy part. F2s are always sibling matings. Alright? Again, not normally done with humans. Okay. But we do that with plants and sometimes with animals to understand genetics. So that means you would take one sibling that was big A, little a, and you would cross it with another sibling from the F1 generation that was big A, little a. And the offspring from that Punnett square equals the F2. Okay? So it's how we label the generations. You've got parental, which is the mating of the parents, which produces the F1. You interbreed F1, and you get the F2. So that is how it's always called. So if you're asked for the F2 generation, that means you have to figure out what the F1s are, and then you have to mate those organisms to produce the F2. Alright, three more slides. Okay, how, excuse me, we represent alleles, right? So we've been talking about big A, little a. And that's pretty easy when we have these single-lettered allele representations, and they're very unique, right? Big A and little a. More correctly, you would represent it as big A slash little a to show the allele pair, to show it's homozygous or heterozygous. And that little slash becomes important when you get to these more complicated types of allele names. Okay, I will accept either way. So you can write this as big A, little a, or you can write this as big A slash little a, and I will know what you mean, right? One of the important things that we'll do, and I'll show you in just a minute, is sometimes you don't know an allele, right? If it's a dominant phenotype, it could be big A, or big A, little a. And the way we represent that is big A with a dash. Okay? Or you can put big A slash with a dash. This means we don't, yet know what that allele is. And that's really important for you to do. Do not assume because something is dominant that it's homozygous or heterozygous until you get more information. When people are doing genetics in the lab, we talk about the wild type allele, which is the most common allele that is being worked with. Okay, and it's designated as a plus. So up here, this is telling you that this is a wild type allele, wild type WT, and that this other allele is actually dominant to the wild type. The point I'm going to make here is that you have to be very careful and accurate when you are representing your alleles. And I'm going to ask you to always define your alleles so that I can understand what you're talking about when you show me work on a genetics problem. Because sometimes there are multiple alleles for a gene. So it's not just one dominant, one recessive. Okay? And a lot of times we use superscripts for that. Right? So we might also have the recessive, little e allele. Okay? You'll see these as we get into more complicated genetics problems and more complicated types of inheritance. Dominant recessive is real simple, easy. As I've said before, not how most genetics happens. You will also sometimes see multiple letters. So instead of just using like a V or something for vestigial wings or C for cinnabar coloring, they'll use a couple letters. And you can now see where the slash would be really helpful, right? So this organism would have the wild type allele and the gene allele for wing type. Okay, so just keep this in mind as we get more complicated genetics problems. This doesn't mean it's two different genes. And that's something I totally forgot to mention when we were talking about assigning alleles. Right? One letter or same letters per gene. Okay? So you cannot say D equals dark and little l equals light. Two different letters or letter combinations would equal two different genes. Okay? So you have to represent one gene with the same letter or the same letter combination. Okay? So don't say D equals dark, and L equals light. Boom, bomb. You lose all your points on genetics problems. Don't do that. It has to be D equals dark and little e, D equals light. Okay? Keep that consistent because then we were know that you're talking about the same gene. Alright. Finally, why am I going through all of this? Because we get to do story problems. Right? And so you'll have lots of story problems. And let's go through this first one to guide you as to what I want you to do to determine outcomes of a genetic cross. So this says, in horses, black is dependent upon a dominant gene B. What I would first do is go B something is black. Okay, you don't know if it's BB, big B, or big B, little b. Chestnut is dependent upon a recessive allele. We know that chestnut has to be little b, because the only way you see a recessive allele, a recessive trait, is if it's homozygous recessive. Trotting walk is due to a dominant gene, so again, trotting is due to big T, little t. Pacing is due to little t. Right? Notice same letters to represent the same genes, the same traits. Always do this. This is what's called define alleles. Okay. If a homozygous black pacer is mated, okay, so let's stop here. We've got a black pacer. Got a black pacer. And it tells me that they're homozygous. That means it tells me that the genetics, the genotype is homozygous. So we've got black and we've got pacer. And then you write an X because that is what mating means, or crossed. To a chestnut, okay, so we know chestnut has to be little b. And it's a homozygous trotter. They tell us that because if they didn't, we wouldn't know if it was big T or big T little t. Okay? Those are the parents. Right? And what you have to do next is figure out the gametes and then do the Punnett square. And this is what we'll go through in class. But I want you to see a couple things. Keep the letters in the same order, or you will get very confused. I also always put a dominant first. So the offspring from this will get a big B from the dad, a little b from the mom, a big T from the mom, a little t from the dad. It doesn't matter who you get your genes, your alleles from. Always put the capital letter first, the lower letter second. Capital letter first, lower letter second. And put them in their allele pairs, right? Put all the B's together, put all the T's together. It will make your life so much easier, and it will make it easier for me to grade and give you all your points when you follow these simple rules. So here's another one. So you might want to pause this video and go ahead and fill this out for yourself. So how I am reading this is that black eyes are dominant to orange eyes. So I'm going to say something is black, and I have to use the same letter. Little b is orange. I guess I should say eyes. And that green skin, I'm going to give it a big G, green skin, is dominant to white skin. So I know white skin is my recessive. I've defined my alleles. So important. Sam, a Mendelian, get it? Mendelian? Ha, okay. With black eyes, so Sam is black and green skin, right? Has a parent with orange eyes and white skin. Why is that important? That's important because it tells us that even though Sam has the dominant phenotypes, he's actually heterozygous because he had to get a little b and a little g from that parent. So he must have got his dominant alleles from the other parent. Really super important. So just because they say black eyes, make that little line until you get more information that tells you what the genotype for that dominant phenotype is. Carol is a Mendelian with orange eyes. So I look back up here. Orange is little b, white skin. Okay, so there's no choices for her genotype. It has to be that way. If Sam and Carol were to mate, predict the phenotypic ratio of their offspring, right? So phenotypic is going to be the description. And we're going to talk about ratios in class, how many of these you get, how many of those you get. All right, I hope this helps you, and I'm excited to work genetics problems with you. And that's it.